Hi students, this is Mrs. Kleins with your next section of video notes over meiosis. You might think meiosis sounds an awful lot like mitosis, so just really quickly I wanted to recap mitosis. Mitosis is the formation of body cells or somatic cells. This is cell division that involves the cells of your body, so skin cells, brain cells, bone cells. Each of our cells in mitosis will have 46 chromosomes. We call those diploid cells or 2N cells because they have 23 pairs of chromosomes. So again, your examples include skin cells, muscle cells, all body or somatic cells. So meiosis is going to be a different process because it involves gametes. Gametes include egg cells and sperm cells, so gametes are another name for reproductive cells. They are used specifically in sexual reproduction. So where mitosis is a form of asexual reproduction, it's one cell that divides into two identical cells. Meiosis is used for sexual reproduction. Each of the gametes will contain 23 chromosomes. Thinking in terms of mitosis, this is half the number of chromosomes in the daughter cells that are produced. During meiosis, we call these cells with half the number of chromosomes haploid cells because they have half the number of chromosomes as our body or somatic cells. We use the designation N for chromosome number, meaning there's just one set where your diploid cells have 2N or are just cells with a full set of chromosomes. You can kind of see that in the picture on the right, there are 46 different chromosomes. There's 2 for each pair. 1 comes from each parent. So during meiosis, what happens is in these diploid cells, the number of chromosomes gets halved. Each gamete that is produced will have just 1 chromosome from each set or each pair. Haploid cells are cells that contain half the number of chromosomes. Gametes are sex cells that have 23 chromosomes. We call it a haploid cell based on its number of chromosomes, and meiosis begins with a diploid parent cell, but the end result is going to be 4 haploid daughter cells. So let's walk through the process and how it occurs. A zygote, you may remember, is a fertilized egg that forms whenever a gamete egg cell and a gamete sperm cell, remember these are both haploid cells, fuse together during fertilization to form the first diploid cell. So in the 46 chromosomes, one set comes from each parent. 46 chromosomes is the normal number of chromosomes. The C is the abbreviation for chromosomes for a human. Okay, in meiosis, there are 2 steps. There needs to be 2 divisions because we're ending up with 4 cells at the end. So the 2 phases are called meiosis 1 and meiosis 2. Another important reason is not just because we're making 4 cells, but because each cell contains half the number of chromosomes as the original parent cell. So first thing, during interphase 1, which is the first stage of meiosis 1, is going to be where the cell spends a lot of its time. And in interphase, remember that there's the synthesis phase, which is where DNA replication takes place so that you've got your chromosomes. They replicate, and now you've got 2 sister chromatids. So we have 1 chromosome. They go through replication. Now we have 2 chromatids that are attached to each other at the centromere. So this is where if you think it sounds a lot like mitosis, you're right so far. Prophase 1, similar to mitosis, we see the nuclear membrane break down. The spindle fibers, if you remember those, also form and they extend out from the centrioles. And what's kind of new or unique to meiosis 1 is the sister chromatids form these homologous pairs with the chromosomes that are the same size and shape. We call these tetrads. Okay, so one of the chromosomes in a tetrad is DNA that came from the father, and one is DNA that came from the mother. They replicated during interphase. When they line up in a tetrad or a homologous pair, they will kind of cross over each other. This crossing over event is how genetic information gets shared among chromosomes. Crossing over occurs when homologous chromosomes or tetrads cross over each other, But when they do that, segments of genes get swapped between the two homologous chromosomes. And this is a very cool way to kind of shuffle up your genes because, as I mentioned earlier, one came from the maternal parent and one came from the paternal parent. Whenever the chromosomes line up in the middle of the cell during metaphase, they are going to be in this homologous pair structure. But before they get pulled apart, they've had the opportunity to share genetic information with each other. So it shuffles up the genetics of humans, which is cool because it increases our genetic variability, and this allows for lots of different combinations of genes and sex cells. That's why siblings are genetically unique from each other. There's not going to be anybody that shares the identical genetic code with another person unless you have identical twins. So after crossing over takes place during the homologous pairing where the tetrads form, they are going to line up in the middle of the cell during metaphase 1. This probably looks somewhat familiar. At this point, like before, spindle fibers attach to the chromosomes at the centromere. During anaphase 1, the homologous pairs get separated. So the homologous pair that formed each tetrad gets pulled apart and they move to opposite ends of the cell. The sister chromatids stay together even though they have at this point already shared or swapped some genetic information with their homologous paired chromosome. Telophase 1 may look somewhat familiar too. Spindle fibers start to break down and the nuclear membranes reform. Cytokinesis 1 still looks very similar to mitosis. The cytoplasm splits equally among the daughter cells. This results in 2 new daughter cells. Now, what's different about meiosis 1 compared to mitosis is the daughter cells are genetically different or unique from each other and from the parent cell. So now those 2 cells are going to enter the next phase of meiosis called meiosis 2. Again, you're going to notice that these are similar to mitosis phases with some slight differences as well. And at the end, we're going to have 4 genetically unique haploid cells. Interphase 2 is a little different from interphase 1 in that during the second interphase, chromosomes do not get replicated. There is no synthesis phase here. The cells just grow and prepare for prophase 2. In prophase 2, the nuclear membrane again breaks down and spindle fibers form. Hopefully, you're starting to see some patterns here. In metaphase 2, the sister chromatids line up at the middle or equator of each cell, and the spindle fibers once again attach to the sister chromatids at the centromere location. I bet you can guess what happens in anaphase 2. The sister chromatids get separated. They move to opposite ends or poles. So each chromatid now is called a chromosome. You've separated these sister chromatids apart. So now each new daughter cell is going to get some different shuffling of chromosomes as the spindle fibers shorten and pull them in opposite directions. During telophase 2, again, the spindle fibers disappear, the nuclear membrane begins to form, and cells start to split apart. You can see in this imagery here the beginnings of 4 new cells. In cytokinesis 2, the 2 cells for meiosis 1 have now divided, resulting with 4 new haploid daughter cells. This is the end goal of meiosis. Each new daughter cell has 23 chromosomes, so these are haploid cells with half the number of chromosomes as a body or somatic cell. Due to crossing over among these homologous pairs and pulling apart during anaphase 1 and anaphase 2, we've got a different shuffling of chromosomes in each cell. Each daughter cell is going to be genetically unique and contains half the number of chromosomes as the original parent cell. Remember that fertilization between a sperm and egg cell is what restores the diploid number by adding these chromosomes together. 23 plus 23 will give you 46, as you can see here. Not all organisms have 46 chromosomes. So it's just important to remember that a diploid cell is going to have double the number of chromosomes as the haploid cell. This image here just shows 3 chromosomes in these haploid cells. But when the sperm cell fertilizes the egg cell, we've got this diploid zygote that has 6 chromosomes. So meiosis occurs in the testes of males. This process is called spermatogenesis, and in the process, one parent cell that starts the process will end up becoming 4 sperm cells. In females, this process will occur in ovaries, and the process is called oogenesis. These 4 cells that are produced are going to be egg cells. So we will review and recap this process together in class, but I hope you found going through the notes that there were some patterns that you observed and some similarities that you observed between meiosis and mitosis, specifically the major events that take place during prophase, metaphase, anaphase, and telophase. Now, I do want you to know what's different or unique about those phases in meiosis compared to mitosis, and how are meiosis 1 and meiosis 2 different from each other, specifically noting during meiosis 1 that the homologous pairs divide, and then during meiosis 2, the sister chromatids divide. This is the end of this section of our video notes.